Okay. Just wanted to say, what a way to end this fantastic conference by just talking about how we end and what happens to us chemically. Yeah, having said that, um, we have a unique odor when we are alive, and beyond our fragrance and our hygiene habits, the antemortem scent or the odor is made up of volatiles that are released from skin through the action of microbiomes, sweat, and the breath VOCs. And so when we die, this kind of our unique odor transitions to what we call as post-mortem odor. And we don't really know when does this transition to the antemortem odor to post-mortem odor happens. And you might tell me, why does it really matter? Because during disaster events such as this, where we are trying to locate victims either dead or alive, one of the methods that are used are the scent detection dogs. And during this mass disaster scenario, we have a search and rescue dog which are trained on the antemortem odor, and they're looking for alive people. And then we have the human remains detection dogs that are trained on the post-mortem odor, and they are trying to locate the deceased victims. And understanding the transition of the odor and at what point this antemortem scent goes away and our profiles look like post-mortem odor helps us to then not only deploy these different types of dog units, but also helps us with training them. And so for the purpose of my research, we looked at the early post-mortem period to see what sort of volatiles are released during this period, and then also track the evolution of the donor VOC profiles to then identify the transition of human scent to post-mortem odor. Before our research, only one study was conducted so far from the pig remains that was looking into the transition of the odor. So before I talk into the experimental design, I will share the early post-mortem decomposition process in humans. So we humans, or decomposition in general, goes through 5 distinct stages. In humans, we see differential decomposition, so more than one stage of decomposition might be occurring at a given point. So we have the fresh stage, the bloat stage, active decay, dry remains, and skeletonization. But for the purpose of my research, I looked at the fresh stage of decomposition, which encompasses the early post-mortem period. Now, during the fresh stage of decomposition, there are the immediate changes that happens in the body, and those are the ulgar mortis, rigor mortis, and liver mortis. So ulgar mortis means the peripheral cooling of the body during the post-mortem period. As the homeostasis stops, our bodies are no longer regulating the temperature. Rigor mortis is the stiffening of the muscles, and then liver mortis is the peripheral pooling of the blood. Now, alongside with these changes, there are also microscopic changes that occur, and these are the autolysis, so the cellular breakdown, and also putrefaction, where this is driven by microbial transformation of our tissues. And so our macromolecules kind of break down and release what we call as VOCs, and they kind of make up the post-mortem odor. So to understand this early post-mortem VOC profile, in our study, we looked at 3 different human donors or cadavers, and we looked at their VOC profiles in the morgue and at the outdoor facility called REST, which is the only facility in Canada that kind of looks into or allows the study of human decomposition in an outdoor environment. And the VOC profiles were collected both at the morgue as well as the outdoor facility to kind of then look into this transition of the odor. Now, bear in mind that the sample collection depends upon the availability of donors. And so as the donor arrive, we kind of collect the samples. Now, just looking at how we collected samples at the morgue and the REST facility. So right after the donors arrive at the morgue, they arrive in a chute-type material. We kind of open this material or the body bag and then let the air out to remove the VOCs that might have accumulated during transportation. Then we kind of reclose the bag again, accumulate the VOCs for 20 minutes, insert the sorbent tube. This is the 10XTA Carbograph 5TD sorbent tube, and then connect the ActiveWalk low flow pump, and then accumulate the VOC, collect the VOCs on top of the sorbent tubes for 10 minutes at the rate of 100 milliliters per minute. We also collect background samples and the samples of the body bags to kind of understand what the background VOC profile is. Moving on, we have at the rest facility, which is the outdoor facility, the donors are unclothed and they are placed on the backs. And then we kind of usually put an anti-scavenging cages to prevent scavenging or prevent animals from getting into the cages. And then at the time of sample collection, we put the aluminum hood on top of them and then let the VOCs accumulate again for 20 minutes and then collect those VOCs on the top of the sorbent tubes and then use the ActiveWalk flow pump to kind of collect the VOCs and then for further analysis. So the methods were consistent between the morgue and the rest facilities and also the analytical parameters. So we used the Pegasus BT for the sample analysis, and the mid-polar combination with the stable bags was used. Moreover, the internal standard was injected in each of the sample, and we also injected the decomposition standard mix, what we call as, it's a mix of 25 compounds that have been previously identified in the human decomposition studies. It's no rival to the century mix, but we use this to kind of track the evolution and how the method performs. In terms of data processing, we use chromatography to kind of do the initial data processing. We kept the peaks that were detected at a minimum of signal to noise ratio 100 and above. And for spectral similarity match, we had criteria of 850 or above. And then the further data processing was done using RStudio. So here we exported our data files along with the mass values, peak tables, as in CSV file. And then we kind of classified the data into the experimental and the control samples. From that point in time, we used a peak pairwise alignment, and then we normalized our samples in terms of our internal standard. Then it was compared with the samples and controls. So any peak that was identified uniquely in the humans samples were kind of retained. Any peak that were identified in background control samples were removed. And then if a peak was identified in samples and controls, then it had to be detected at a signal to noise ratio in the area twice and to be considered. Then we also filtered these peaks and removed any background contaminants or siloxanes coming from the columns. And then we were kind of merged the individual data files into one big Excel files containing the normalized peak areas. From that point in time, we ended up finally with a list of compounds with the normalized peak areas. And then again, we went back to kind of manually verify those peaks, go back to the chromatography software, and also look at PubChem libraries to just look if the candidates were identified correctly. So this kind of finally leads us to the results. Looking at just the decomposition patterns, within just the 3 donors, we had different initial patterns of decomposition across the 3 donors. So these are the patterns that are seen usually as a result of decomposition processes. And just within the same environment, the bodies kind of decompose differently, and no 2 bodies in our experience we have seen that kind of progress similarly in terms of decomposition process. And so this kind of just shows us how variable the samples could be and the progression of decomposition within the same environment. In terms of the early postmortem VOC profiles at the rest facility, we saw a mix of chemical classes that were identified across the 3 donors. And amongst them, the nitrogen-containing compounds had the higher abundance, whereas the acids had the lower abundance. And in terms of detecting acids, usually they are detected in the middle and the later phases of decomposition. So it was kind of what we saw in the overall decomposition profile in the literature. Now, to track the transition of the odor, we kind of combined the VOCs that were detected in the morgue and the rest facility, made a data set out of it, and then we looked into the literature, into the studies that were looking at antemortem VOCs from a mass disaster perspective, and also compared it with, compared the database with the post-mortem VOCs that were previously detected from human remains, especially human remains. And then we later on identified 24 VOCs or 24 antemortem VOCs that were kind of common with literature, and then 35 VOCs that were common with the decomposition literature or postmortem VOCs. One of the interesting things that we find was that there was a group of VOCs that were identified both in the ante and the postmortem period, and in literature, these VOCs were kind of identified in both the postmortem and the antemortem period. In the antemortem period, they were identified from skin matrix, and in the postmortem period, they were linked with microbial breakdown of macromolecules. And so we kind of classified them as its own separate class, and we call them as transmortem VOCs. And so what we could see is that this kind of allowed us to better understand the progression and the change of the VOC profiles during the early postmortem period, and also understand the data better. Now, from that point in time, I would like to show you the VOC profile results of different donors. So the x-axis shows us the ADD, and this is the accumulated degree days. So we collected temperature data as soon as the bodies were placed at the rest facility. And so ADD is just the sum of the averages of the temperature. And then what we could see is that the antemortem VOCs were detected on each experimental day, even during the early postmortem period, as well as the transmortem VOCs were detected across the samples. But the interesting thing was that the amount of postmortem VOCs just increased during the later stages of the experimental trial. And I'll get into why we saw that. Now, in this instance, we could not collect samples on ED3 or the third day of the experimental trial, and that was just because of the rain that happened on that day, and we couldn't really collect the sample. Now, this is where the presentation stops. Okay. For donor number 8, we also saw a similar trend where we had the antemortem VOCs along with the transmortem VOCs that were being detected, and then the sudden increase of the postmortem VOCs from ED3 day onwards. And if we move on to donor number 9, then again a similar trend was seen where the antemortem VOCs were present or identified throughout the trial. We had the transmortem VOCs, but there was a sudden increase of the postmortem VOCs at the later stages of the trial. So visually, on ED3 day onwards, we started to see that the bodies showed signs or progression towards the bloat stage of decomposition, and the profiles kind of changing. And so we could say that just with the visual confirmation and also looking at the VOC profiles, that this transition of the odor starts around ED3 day onwards, where the visually as well as chemically, we start seeing the abundance of postmortem VOCs becoming higher. So also the presence of antemortem VOCs and their persistent across the early postmortem stages kind of led us to believe that this is what could cause, this is what it could cause the confusion or at times the challenges that the scent detection dog faces in the field in kind of locating alive victims. We have seen and talked with the dog handlers where they have reported that at times the search and rescue dogs kind of alert to the deceased victims. And we think that because of the persistence of the antemortem period, antemortem and transmortem VOCs, it could kind of lead to the challenges that the dogs might face in the field. Also, the postmortem VOCs became more prominent as the decomposition stage progressed, and we could see a transition of the odor in humans in an outdoor environment. And this is kind of big for us because we were never able to see or detect this transition when I did this study in Australia way back in 2016. So what I believe is that it's just a start, or we are just scratching the surface, and lot more research needs to be done in terms of looking at the odor transitions in different environments and also across different seasons. And so with that, I would like to acknowledge the donors who kind of agreed to donate their bodies for this kind of research. Without their donations, it would not have been possible to do this research. Shali Durgan, who is also was my lab mate during my PhD and kind of has helped me with a lot of sample collection, standing outdoors. Forbes Research Group, the multidimensional gas chromatography community. Thank you for always answering the questions that I had, and different funding agencies to make this trip possible, and the research. Thank you.